Okay. So thank you everyone. Sorry for this short delay in. Okay, so I will very briefly introduce Eric to our audience. So Eric is a biologist, microbiologist, evolutionary biologist by training. He has also one specificity that is of interest to our audience today, which is that he has also a PhD in philosophy of biology in particular. He's been also I'm saying that in passing, for those of you who are curious, you can just look on his webpage. He has also published papers that we would call philosophy in science, typical examples that we examined with Thomas when we wrote the BJPS paper on philosophy in science. More recently, and you will see now, he has started to develop an interest in aging, and is, as far I don't know the content of the presentation. I think I know the paper on which it's based, and it's, as you will see, always a lot of original ideas. That's really a typical hallmark of Eric's presentation. You have 3 new ideas per minute. But he's a bit under the weather today, I think. So maybe it will be less new ideas, maybe just 1 per minute. Okay, you will probably, yeah, I guess we have an apology from you for that. Yeah. Okay, so Eric, the floor is yours.
Thank you very much. Thank you very much for inviting me. Thank you very much, Maël. Thank you very much, Thomas. So I will be essentially telling you about the content of my new book that is in English called The Elastic Life, Revolutions in Aging, ? But in French, it's called a bit differently. So Thomas, to move to the next slide. So this talk will be at the interface of biology of aging, theoretical biology, and evolutionary biology. And it will be consideration on the current theories on aging and how they have some gray areas in terms of knowledge about development, phylogenetics, eco-evolutionary interactions, systemics, and some ontological issues, and how these gray areas are actually opening room for many new questions about aging, including philosophical questions. So I will first start with a disclaimer. That is, this is super fascinating to study aging, but I'm unable to tell you about everything that I find fascinating about aging. So in this talk, it will already be very dense. You will hear a lot of things. Some of them may interest you, but I will not cover every topic. Some of them will be for another story. So if we start by imagining that we want to address what is seemingly a simple question. Do we know whether chimpanzee, yes, we know that. Do we know why chimpanzee age in a different way than humans? To address this question, we need to explain aging. And for this, we can be quite demanding in what we would like our explanation to cover. For example, I would like my explanation to offer me information about the proximal causes of aging, and I will come back to that later, the ultimate causes of aging, and also the natural history of aging, how the various mechanisms that are involved in aging came to be. And so, just so that we're on the same page, what I would welcome as an explanation for aging would be something about the how question. What are the mechanisms and processes that are responsible for aging? And some hints may be found in this famous hallmarks of aging that you see on this slide. There may be possible causes of aging, also nobody knows about this. There may also be consequences of aging. I would like also to know something about the why question, what selective pressures led to the evolution of aging. And like I was saying, I would like to know something about the natural history of this mechanism and process. So if you imagine that, I don't know how to point with this system, that the little networks that you see on the top of the contemporary human being are networks associated with aging today. So for example, interaction between proteins that contribute to the aging of the human today. We would like to know how this network came to be, because it comes from a long history and it has a past that we would like to uncover. So if we have all of this, proximal explanation, ultimate explanation, natural history, I think we are in good shape to understand aging in a decent way. But the problem is that most of the theories, they focus either on proximal explanations or on ultimate explanation, and they are rarely complete. So for example, theories based on mechanism offer incomplete explanation of aging, and that is pretty clear if you take the case of wear and tear, the accumulation of damages during life that could be involved in aging. If you explain aging in this way, this is incomplete first because you also need to consider not just what is breaking the organism down, but also what is repairing the organism, because organisms that just don't get more and more damages in their bodies, they also have some ways that are evolved ways to fight against these issues. And so for a certain period of its life, an organism seems to be able to repair damages quite well. But for another period of its life, then the damages seems to be accumulating faster than they are repaired, or maybe even repair are stopping. To understand aging, we need to take into account two kinds of forces that are opposite, the ones that break down the organism and the ones that repair the organism. And if we know how to do this, how to explain this, then we can say maybe how we go from one period of life where an organism can grow and develop and become sexually mature, and another period of life where the organism seems to be doing less well. But again, if we look at this kind of explanation, we can find many mechanisms that are breaking down the organism, from internal mechanisms like protein being degraded, DNA accumulating trouble, and also external mechanism. Maybe you have an injury in your environment, and all of these mechanisms together will not be sufficient to fully account for aging, because, like I said, at some point, we also want to know something more. We want to know not only how, but why. Why is it the case that during the life of an organism, at some point, repair becomes less efficient? Or maybe the organism stops repairing itself. And this why question leads us to the universe of the ultimate causes to explain aging. And there are also theories that now mostly focus on ultimate causes that have been put forward to explain aging. Those are evolutionary theories of aging, and they also present gray areas. The main ultimate cause that is invoked by these theories is called selection shadow. Selection shadow is the fact that past a certain age, organisms are no longer seen by natural selection. And this is expected because before their fertility peak, whatever is affecting the organism can have dramatic consequences in terms of fitness. For example, if you have a terrible disease before you reproduce, then you will never transmit these genes that were giving you this terrible disease, and it will be strongly counter-selected. But if you have a terrible disease after you have reproduced, well, it will not make on an evolutionary perspective much of a difference. It may be terrible for you, but you would already have produced your offspring. And so natural selection will, in a sense, not see all the terrible things that are happening to the organism past, and especially a long time after its peak of fertility. And this selection shadow is an admitted explanation for the mainstream evolutionary theories of aging. And precisely in this way, the theory of mutation accumulation explains aging by proposing that there are some late deleterious gene expression that will impair the organism, but in a sense too late to be counteracted by natural selection. And so an organism that will have its terrible genes acting on later in his life will have already transmitted this gene to its descendant. And so in that sense, the genes with late deleterious expression may accumulate in lineages. And that is proposed to be a cause, an ultimate cause of aging. Another explanation that also consider that selection is stronger early in life and less efficient late in life is antagonistic pleiotropy. This explanation of aging insists on the idea that there are some genes that may do you good when you're young, even though the same genes make you trouble, cause trouble in your body when you're older. These kind of genes, if they favor your reproduction when you're young, will be favored by natural selection. They will be in your lineage, even though as a collateral damage, they provoke later in your life all sorts of problems, all sorts of physiological issue, and contribute to your aging. So the example for this is this horns of the babirusa where they have very sexy horns, if you wish, when they are young males, but later on in their life, these horns would pierce their skin and they would get infected, and eventually it could even crush their skull. So there are some things that are good early on and bad later on, and because of the good things are more strongly selected early in life, then you end up aging late in your life. And the third theory is called the disposable soma theory. So it considers that there are two important compartments in the organismal body, the germline, that is devoted to the reproduction of the lineage, and the soma, that is the rest of the body. And in this theory, the key notion is the notion of a trade-off, energetic trade-off. Energy goes first to good maintenance of the germline and at the expense of the maintenance and repair of the soma. This makes sense if you are little mice, for example, living in a very dangerous environment where you're going to be predated upon and you will die young no matter what. So there is no point in creating a very strong, ever-living body for a mouse. But there is a lot of sense to try to invest your energy to reproduce fast before you get killed. And so in this kind of theories, natural selection would favor reproduction at the expense of the survival of the body. And because damage will then accumulate in the soma, there will be some aging. So all these theories are the mainstream evolutionary theories of aging, and they share some important elements, but also some important blind spots. First, they are centered on organisms that are presumed to be aging in population from a single species. The environmental and ecological conditions are at best in a black box. These theories are meant to be applied to eukaryotes, if not even to animals. And the three of them agree that aging is non-adaptive in the sense that aging is not selected for itself. In the first theory, mutation accumulation, aging is a form of evolution neglect, if you wish. Selection shadow makes it impossible to prevent aging, but it's not that. Leadbeater's expression is for aging. Antagonistic pleiotropy, likewise, is all about optimizing reproduction, not optimizing aging. And disposable soma theory is also intended to allow the organism to better reproduce, not in order that the organism age. So having said this, if we go back to my first question, can we explain why chimpanzees are aging in a different way than humans? The answer seems to be no. Even if we conjugate the knowledge on the mechanism of aging, so the proximate causes of aging, with the theories with the ultimate causes of aging, we cannot really predict why our two species age in a different way. And that is because this framework is incomplete and these theories have some blind spots. So can we do better? I think there is an epistemic strategy we can try to use. That is first to identify the gray areas of the theories, dive in these gray areas, and try to bring in some new lights to try to provide additional explanations of aging. And in order to convince you that there are such gray areas, I will go through a series of examples of what are important features of the theories that basically we know too little about and that maybe we should bring forward in our model. So first, these evolutionary theories of aging present some gray areas about the development. They all assume that somehow there is some horology in the organism. That is, gene expression that will be either early or late, or earlier or later. And in a way, they assume that there are clocks within organisms that are structuring, regulating as well gene expression order, and eventually during the entire lifespan of the organism. That is true for the non-adaptive theories of aging, but that is also true, obviously, for theories of aging that would be invoking programs for aging, because these programs would have to be instantiated at specific time. It is true as well for a trade-off. When do you start to give up the soma? When do you start to favor the germline? And the biologists who are the most advanced in studying clocks in organisms, I believe, are not biogerontologists, but they are developmental biologists who are trying to uncover some programs of developments. And so maybe the study of aging has something to learn from the study of development. But that can only be done by making the horology issue explicit. We all assume that organisms know what age they have in a sense, because otherwise, how is it possible to know what gene will be firing before what other gene? But when we look in the biological knowledge and we try to identify what those clocks are, then we face a deep biological mystery. In fact, one of the deepest, because we simply don't know what is the ecology of the organism. And it would be a big challenge for all these theories to try to uncover how the organism knows what time it is and what age it has. And that would bring a lot of new knowledge to have this ecology at the center of studies on aging. First, because if we can uncover clocks that runs for the entire life of an organism, then that will contribute to the debate on the programmed or not programmed nature of aging. For example, it's difficult to have a program aging if you don't have a clock that runs throughout the entire life of an organism. But that's beyond the scope of my talk. And also, this suggests that if we find that there are some clocks that are able to measure the time that's passing by some organism, and we find those clocks in many more organisms than we are currently investigating aging in, then we may suspect that there would be more entities and more organisms aging on Earth than what we currently imagine. If the clocks are needed to age and we find the clocks everywhere, then maybe aging is everywhere. And that's a second kind of example I would like to discuss about the evolutionary theories of aging, how they present some phylogenetic gray area. That is, how they apply to a part of the phylogenetic tree of life, but probably not sufficiently deep in this tree. So let's take the case of the disposable soma theory with this neat German and soma distinction that is at the heart of the explanation of aging. This kind of German and soma distinction is observed only in animals. And that would mean that this theory cannot go in terms of explaining the evolution of aging deeper than the origin of animals, and even animals with German and soma distinction. And that actually is the case for a reason. This is that this theory is not trying to explain the natural history of how germlats and soma came to be, and how eventually trade-off could be made, what mechanism could evolve and did evolve to divert energy towards the germline rather than towards the soma. However, if we would like to put this kind of question more central stage in the question about how aging evolved, then it would be fascinating to do, because if aging is related to the evolution of germ and soma, then to explain the evolution of germ and soma, we need to solve another deep biological mystery. That is the origin of animals and the origin of germ and soma. And this question is very, very difficult to address. But we can hardly explain the origin of aging without explaining the origin of animals. And that is a typical major evolutionary transition during evolution. So it would seem that our evolutionary theories of aging should include something about major evolutionary transition and aging. For example, the unicellular ancestors before the first animals could have been aging as well. And so if these cells were aging, then aging would be more ancient than animal aging. And in this sense, the theory of disposable soma will not go deep enough, because aging would predate what is meant to explain the aging in animals. This is probably the case, that aging is older than animals, and so the theory could be expanded deeper in the tree of life, because when you look at the various mechanisms that are associated with aging in mammals, what you do see is that some of these mechanisms are present in other species that are branching outside the animals. And so if I could be pointing somewhere on this slide, but I cannot do it, outside the yellow part. I'm scared that it will actually go to the next. No, it doesn't show on the screen. Well, just for you, but it doesn't for me. So it's a bit complicated. I'm not smart enough to do this. So I will go with Word. What I would like to say is that there are important features such as, do you stop your telomerases? Do you fix your DNA? Do you repair your proteins? That certainly evolved before animal evolved. And in that sense, there are quite a few mechanisms that suggest that aging is old, and certainly older than animals. And there are experimental evidence that shows that unicellular organisms, some of them actually do age biologically, meaning that they have functions that are less and less effective with time, and that they do age replicatively, meaning that they are less and less able to reproduce. But that is not modeled by the standard evolutionary theories, aging in microbes. Is however observed, for example, for the bacteria Colobacter crescentus, that has an interesting life cycle where first a swarming cell is going to fix on a substrate, and then on this substrate, the cell is now stuck, but it will divide, and the bottom part of this dividing cell will stay there stuck on the ground, but the upper part, it's a new swarmer that is freed and that can restart the cycle. Interestingly, if microbes were not aging, then the bottom cell could endlessly divide and give birth ever and ever to new daughter cells, if you wish. But that is not the case. And in fact, there is a limited number of cell division that is possible for the bottom cell that suggests reproductive aging. Likewise, for E. coli, it has been proposed that these cells, also that divide very symmetrically, would actually be aging because of the age of their poles. And to understand this, you need to look at my hands now. So this is an E. coli, and it's dividing. To divide, it must first make a hole in the membrane, and it must create new membrane to close the new cells. This new membrane here is younger than this old membrane on the other side of the bacteria. But if you imagine the process goes on again, and this side will be very old with respect to the rest of the bacterial membrane. So there is what is called old membrane poles and young membrane poles in E. coli, and the cells with older membrane poles seems to be doing less well in competition, and they seem to have at least biologically aged. In the yeast Saccharomyces cerevisiae, which is another unicellular eukaryote this time, there is also many observations of aging for this yeast. For example, you see here a dividing yeast that is budding a new born cell, and each time this yeast cell is budding a new cell, it has a scar on its surface, and those scars can be counted, and the number of times the yeast cell divides can also be estimated, and it is not indefinite, it is limited. There is a certain number of cell budding that is possible, and that speaks of reproductive aging of this unicellular organism. And so there are now models for explaining aging in single cells. If you start to the bottom left of the slide, what you see is that there are cells that can accumulate damages in their cytoplasm, on their proteins, in their DNA, on their old membrane pole eventually. And what is critical in this model is that one way that this lineage that is aging and accumulating trouble is not disappearing is that at some point during cell division, there is an asymmetric process that puts most of the damages in one of the two daughter cells, and this daughter cell with lots of damages is sort of aged, whereas the other daughter cell has less of the damages, and in this sense seems to be rejuvenated. This is interesting because in these models, what is critical to explain how cells that are unicellular can both age and not disappear because their lineage otherwise would be doomed to failure if they were not able to rejuvenate, is precisely that they rejuvenate by asymmetric cell division, with some of the cells sometimes being able to get rid of some damages that were previously accumulated in their lineage. This is in a contrast with what Weissman's seminal work on evolution of aging suggested, where Weissman was proposing that when a unicellular divides, it divides symmetrically, giving birth to two daughter cells that are exactly equivalent. And in fact, there is no dead body, no cadaver. So this kind of life could go endlessly unless there is predation and this kind of thing. But he was foreseeing that there is immortality, if you wish, for the unicellular organism. But nowadays, biogerontologists consider rather that whenever there is asymmetry, it's possible to have some labor division, and in particular, when there is asymmetric cell division, it's possible to generate one of the cells that has accumulated trouble, and that looks like a transient soma, if you wish, accumulating trouble, and another cell that seems to be rejuvenated, like a transient germen, if you prefer. And this is outside the scope of this talk, but I've proposed a hypothesis that says that apparently there is no way to have anyway a symmetric cell division because DNA replication is always semi-conservative. And for that reason, each time a cell divides, the two resulting cells will inherit DNA strands of different age. Not the same strand will have the same age in the two daughter cells, so there will be a universal asymmetry. So maybe there is room, because there is a symmetry, for some labor division around the aging and rejuvenation to evolve since the first cells evolved. But beyond that principle that I'm claiming, and you could read the paper about this, what I want to say is that we know really little about microbial aging within the tree of life. We have examples for a few bacteria and for a few protists, and we know in particular absolutely nothing for whether archaea age, and archaea are a big part of the evolutionary history. They are this third domain of life, presumably closely related to eukaryotes. Among the things we don't know much about the demographic aging outside eukaryotes. Demographic aging, to remind you, is the story that once organisms are adult, we can compute the risk of dying with time. Is it the case that the risk of dying of adult organism is increasing, as in our species? Then we say it's a senescent species. Is it the case that the risk of dying of adult is decreasing with time? This is the case of many turtle species, and we say that they have a negligible aging. Or is it the case that, like Hydra, the risk of dying is not changing with time, and so we say these species are not senescent? So studies are made by biodemographers on several species of eukaryotes. But where would microbes sit on such a plot? It's very difficult to tell because, in fact, first, it's difficult to know what would be an adult microbe. Would you say that a microbe is an adult once it has divided or once it has replicated its DNA? Or maybe the analogy doesn't hold truly. But it would be interesting to know along a cell cycle whether the risk of dying of cells is actually increasing, decreasing, or stable. And so we partnered with excellent biodemographer Annette Baudisch, an expert in aging, Claudio Franceschi, and we decided to try to measure the risk of dying of cells from the archaeal domain, the one for which we know nothing about. And for this, we studied a species of Saccharolobus. We needed to collaborate with excellent experimentalists from the Pasteur Institute, Mark Krupovich, Virginia Krupovich, and Miguel Gomez Raya Villanova, who basically grew synchronized culture of these archaeal cells that are growing at 80 degrees. So it's very difficult to do. And once they had synchronized the cell, what they did is basically they counted within the cell population as the cell cycle was moving forward, whether cells were dying increasingly or not. And so they counted the live cells in green and the dead cells in red. And along the cell cycle, so from a phase where there is one cell with two DNA molecules, and then the two DNA molecule get ready for cell division. Then there is cell division, it's called DG1. And then after this, there is cell division. There are now two cells each with its DNA molecule. And so these cells continue growing, and eventually they replicate. And there are now two DNA molecules in the cell, and then they get ready for the next division. That's the cell cycle of Saccharolobus cells. They counted how many dead cells, how many live cells. And you may see on this plot that at the time of cell division, there is more dead cells than in the rest of the cell cycle. So basically, since the time of the cell division, what happened for Saccharolobus is that their risk of dying reduces. So if you imagine that these cells after cell division were already adult, then they would seem to be negatively aging. If you imagine that they were not adult, but they only become adult before they divide, then there would be a form of pre-reproductive mortality decline or ontogenescence. This is super cool because, no, we still know very little about microbial aging within the tree of life. We know a bit more about aging in one archaeal species and still very little about the rest. But we know even less about aging beyond the tree of life. And that is a very important point because cells are not the only organism evolving with genetic information in them. There are also viruses and plasmids that are evolving in this way. And if you look at these figures, the network to the right, you see where the cellular life is present. This is a green node, and you see the rest of the universe of what is evolving and has genetic information. And so it is interesting because in this rest of the universe, there are many, many viruses that play a critical role in biogeochemical cycles, and they have been forever around on the planet. And it's very unusual to ask whether these viruses themselves could be aging. So we said before, it's very possible that aging is old, and we need to expand the phylogenetic scope of theories to see this aging in a bigger evolutionary picture. But if we want the real big evolutionary picture, we could also go beyond cells and see whether they are aging. So that's also beyond the scope of this talk, but I committed a paper that proposed a hypothesis to explain how some viruses may be aging. And that is based on an excellent paper that is by Duffield and Demagales, who indicated that when there is an epigenetic system that is responsible for the functioning of what they call higher organisms, so mammals, and this epigenetic system has trouble, but cannot fix the trouble, then higher organism with an epigenetic system that cannot be reset will inexorably age. And it occurred to me that actually microbes and even viruses, some of them really use an epigenetic system to complete their life cycle, if you wish, to replicate in particular. And so if the viruses don't know how to fix the issues that are happening to the epigenetic system, they may also age, these viruses in particular. But beyond this hypothesis, again, there is something more important to consider. Why would it matter to imagine that microbes are aging? First, it would matter because it would expand the scope, needed scope, of the explanation of aging, not only within the tree of life, but also beyond the tree of life. It would provide us with a much needed big picture of the evolution of aging. It would provide us with new models to study aging that are these microbes. For example, we could study aging and rejuvenation in archaea. We could study maybe aging in viruses and know maybe if they counteract aging, anti-aging recipes. And we could also investigate all these pandemics and ecological interaction in a very different way. There is something to be learned about eco-evolutionary dynamics if you consider that old viruses do not do exactly the same thing when they infect you than young viruses, or if your microbiome is loaded with old bacteria rather than with young bacteria. So it's possible that there is a field to be uncovered there. And in addition, there is a big ontological challenge, because if we assume that asymmetry in cell division is needed for rejuvenating single cells when they are aging, that it seems impossible that a single cell truly exists and be a single cell for its entire cell cycle. Because at some point, it needs to be bicellular. It needs at least one garbage cell. And that is this way that the clone can keep going on. So maybe what we assumed before, that there would be microbes, they would be unicellular, is not consistent with the idea that microbes age. And maybe we need to imagine that microbes are at least bicellular, even if transiently, rather than unicellular. Now I move on to a third kind of gray areas that are in these evolutionary theories of aging, and they also can be apprehended by consideration on microbes. These are eco-evolutionary gray areas. I will give you two examples of this. First, example of age distorters, which are entities that manipulate the aging of members of other species, and I will go at length on this. And then I will tell you about not selection shadow, but host selection shadow. And that would be fun to listen to. So first, age distorters. If you remember my picture of these processes associated with aging that evolved over time and eventually led to some interactions that in present-day organisms contribute to their aging, then we can wonder whether some organism, some maybe viruses around us, could not be able to graft their own genetics onto these processes, onto the host processes, and somehow manipulate it. The reason why we are compelled to ask this question is because we believe that organisms emerge from molecular networks, so interaction at the molecular level, and cellular networks and organ networks, but we also believe that these organisms emerging from networks belong to, are surrounded by ecological networks, or social networks, if you wish. They belong to communities. And it has been a long time, actually, that we have been surrounded by microbes that, in the case of the microbiome, outnumber our human cells, and that we have been surrounded by viruses and parasites. But it was also true for our ancestors. They also were exposed to a lot of interactions throughout their evolutionary history with microbes. And so that means that there have been a lot of time for many species whose life cycle intersect with our own life cycle to learn how to integrate some of their genetic devices into our own networks. And typically viruses that do this, when they infect a cell, they would graft their viral protein onto the host cellular machinery and take control of the host cellular machinery so that the cells will now reproduce viruses. And so that's not science fiction, the notion that we own our body to the extent that we don't share it with other members of other species, but at some point we share our body with other species, for example, when they infect us. And this is interesting because in terms of the origin of aging, it offers an alternative possibility to explain aging. We tend to believe that there are genetic causes of aging possibly inside the organism, genes that contribute to less effective repair system, or eventually genes that contribute to the aging of the aging organism, but they are the genes from the genome of the aging organism. And we also understand that the environment causes a lot of aging, but what is possibly underappreciated is whether there are some genetic causes of aging that come from the environment, from outside the aging body, when two life cycles intersect. So this is a representation of this. In blue, you have a bit of a network of the host that leads to the host aging, with blue being, for example, human proteins associated with aging. And in red, you have these proteins now coming from candidate age distorters that can interfere with the normal unfolding of the host pathway. And so what you see here is that maybe the red protein are slowing down host aging or accelerating host aging, but they might interfere with it. Do age distorters exist? The good candidate to look for them would be in viruses, pathogenic bacteria, parasites, symbionts, and even in our microbiomes. And yes, there seems to be such a thing as age distorters in nature. For example, in the case of the nematode Hymenelepis diminuta, it's a nematode, so it's a worm. It has a complex life cycle because it must first infect a beetle, and then the beetle must be eaten by a rodent. And the nematode can only complete its life cycle when it is in a rodent. So this nematode actually is able to enhance the lifespan, make beetles live longer, because in that way, beetles have more chances to be eaten by rodents. So parasite with complex life cycle, an intermediate host can manipulate the lifespan, the longevity of the intermediate host. There are also examples of bacteria in nematodes that basically makes the life of the nematode shorter or longer. So the genes in the bacteria are actually critical to explain the lifespan of the nematode host of microbes. And there are, also it is debated, observations that some viruses could accelerate human aging. In the case of HIV-1, it has been proposed that various phenotypes that are described as aging are observed in patients that are infected by HIV-1, and it has been measured independent of treatment, and even in the first, early after the infection, that the epigenetic clock would be accelerating because of HIV-1 infection. So what we did in the lab is actually we look at all the genomes of all the viruses to know, for those that had tested, experiment tested protein interaction with human proteins, whether they were viral proteins that were able to interact with human proteins associated with aging. And the human proteins associated with aging are the center of this graph. And what you see around is that there are 69 human proteins that could be targeted by a diversity of viruses. So when we get infected by these viruses, it is possible that the way we age is somewhat altered. We did the same game for microbes, for which there are bacteria, for which there are less data. And we also identified some gerogenic bacteria, so bacteria that would have consequences affecting, altering, distorting our aging. And we found that they, in particular, within the 69 human proteins, target 14 human proteins that may be Achilles' heels in terms of aging in the face of infection. So protein in humans associated with aging that eventually are going to be connected and interfered by microbial proteins. So if that was the case, then our aging would be dependent from our environment, fine. It would be dependent from our genes, that's fine too, but maybe also from genes of other entities that use our body for their own interest. If you think, what is the interest of viruses to have us aging, for example, it may be because with a weaker immune system, these viruses could reproduce better. But this age distortion is certainly not modeled in current evolutionary theories of aging, and at most, current theories of aging consider microbiome dysbiosis as a microbial possible cause of aging. So I think that there are some viruses we suspect to be altering or staging, but maybe there are many more. Maybe that's just the tip of the iceberg, and there are more age distorters to be uncovered. If that were the case, that would be especially interesting because it would expand the kind of theory we do about aging. Classically, we talk about the evolution of aging. That is, we focus on organisms from a given species. But if aging is also an interspecific issue, then we could expand our theories to co-evolution of aging. And then it would involve multiple species at once. This would be an extension of the classic theories in many fun ways. So if you remember the disposable soma theory, on this plot, you see a little ghost. The little ghost is when the soma gets disposed of so that the germline can give birth to gametes that continue the lineage. That's the classic theory of disposable soma. But now if the organism, its life cycle is intersected with an age distorter in blue here, then the age distorter may decide in part what portion of the host body are to be disposed of. And eventually an age distorter may dispose of the soma and the germline. So what is fun here is that when you meet an age distorter, eventually it's not just your soma that could be disposed of, but your germen as well. And that is because it's very possible that some, at least in theory, sorry, that some aging genes could have evolved in order to age an organism, but not within an organism, because within an organism, the theory says aging is not adaptive, but aging genes could have evolved in enemies, if you wish, of the aging organism to get it to age. That would make a theoretical option where aging could be adaptive, but not for the one entity that is aging. And likewise, we could imagine to expand the antagonistic pleiotropic theory by considering age distorters. There would be interaction with some viruses that do us good when we're young, but harm us later in our life, for example. So if we really take this idea seriously, that it's possible that the way we age depends on what other entity we meet and whether these entities have some potential to accelerate or slow down our aging, then it means that we must make room in our theories for aging of infectious origins. And because this infection comes from our environment, maybe we need to develop some sort of biosocial aging concept, which we are doing in collaboration with Philippe Unman and with Juliette Garzinski in my lab. The second example of interspecific interaction that are not modeled in mainstream evolutionary theories of aging and theories of aging, but could be modeled in it, is what I call host selection shadow. So we know that a dysbiosis, a disequilibrium of microbiomes, is going to affect the host body. This is a red arrow, in the sense that microbiome, when they're in dysbiosis, would result in some chronic inflammation, inflammaging, that is actually involved potentially in almost all age-related diseases. But we know far less of the impact, the orange arrow, the impact of the aging host on the microbiomes. And this is a theoretical interesting point, however, because if you think of the relationship between a host and its microbiome, there is a model that's called ecosystem on a leash that depicts it in a neat way. The host, and you see the red arrows going from the host, exerts some control, some selective pressure on its microbiome. That's at the top of the polar bear. The red arrow says, for example, that the immune system of the host is going to contribute to shape the microbiome. But that model is for an optimally functioning host, if you wish, an adult that is healthy. But if you remember, because of selection shadow, according to the mainstream theories, this optimal functioning of the organism past its fertility peak is probably going to be reduced. And so if aging is altering some important host function, and in particular the host immune system, it is possible that selection shadow is going to interfere, affect the way the host is selecting its microbiomes. And it's possible that the host is now exerting a different and possibly a lesser control on its microbiome. So if that is the case, what we are observing in this theory is that selection shadow has a replicate that I would call host selection shadow, because natural selection doesn't see the host as well as when it was younger. The host do not see its microbes as well as when it was younger. And if that is the case, if there is this kind of host selection shadow, then what selection shadow would do for host genes, typically that is not being able to get rid of problematic genes late in life, could also be replicated at the level of the microbiome. It could be the case that host selection shadow is actually resulting in some accumulation in the microbiome of terrible genes, late microbial genes with terrible expression, or even genes with antagonistic pleiotropic effect. But what I wanted to stress on with this notion of host selection shadow is that the mainstream theory focuses on the aging of a single species. But here, we need to consider aging across scales. The scale of the microbiome, the scale of the host, and those are different scales with not only different spatial scales, but also different time scales for evolution. And we need also to consider aging through systems in interaction, because if the way the host ages affect the way microbiomes behave, which affect the way the host ages, then we need to shift from models that are inspired by organismal metaphor to explain aging to models that are more systemic in nature. So why does this interspecific aging matter? Because it calls for modeling aging in a way that is multi-scale, because it expands aging now beyond its usual targets, not only in the tree of life, but in the web of life. And it compels us to think about co-evolutionary dynamics of aging and co-evolutionary theories of aging. And there are new concepts that are needed to do this, such as host selection shadow, arm races with age distorters, infectious aging, and something very strange I will tell you next, XD Darwinification. But to do this, we need to go through almost the last two parts of my talk, and there would be additional gray areas that I think are problematic for the mainstream theories of aging, systemic gray areas and ontological gray areas. And I will start with the ontological ones, the ontological gray areas, because it is obvious that aging is actually a kind of a stress test for identity. This is now the depiction of a different theory of aging that is also quite prominent, also much more recent. It's called the information theory of aging. In this theory, epigenetic is very important. What epigenetic does is that it changes stem cells into differentiated cells because of some epigenetic signal within the cell. For example, you start from a stem cell and the epigenetic specify it will become an eye cell or a brain cell or a muscle cell. And that's fine, because this is then going to be functional in the organism. But as time passes or some injuries happen, or de Magalhães would say inexorably, the epigenetic signal will get lost with age. And that means that what was creating and maintaining cell identity will also get lost with aging. And so the differentiated cells, the cells that were now muscle cells, will turn into something different. They will no longer be muscle cells with age. They will turn into another kind of cells. They will X-differentiate. That's the terminology by David Sinclair, who put up this theory. So that's for the start of my story. That at the cellular level, at least, there could be something called X-differentiation, that is a shift, a change in identity of the cell during aging. And all the work by David Sinclair is to try to redifferentiate those cells so that they don't go too far off what they were supposed to be doing and they go back to the status of muscle cells, for example. But what I want to ask is a more general, deeper, also connected question, and that is the following. What is happening to Darwinian individuals during aging, not just to cells in one organism, in ontological terms? So what are Darwinian individuals? They are those units of selection that, if you wish, natural selection sees, and very smart people have proposed, for example, that among these Darwinian individuals, you could have an organism, for sure, a virus, a gene, and a social insect colony. And I'm not going to discuss whether this is the right boundaries for what Darwinian individuals are. What I want to stress on is that, very importantly, Darwinian individuals could be composite of many components that underwent a very specific process that's called de-Darwinification. So if you look at the plot by Godfrey Smith on the left here, and you have two Darwinian individuals, for example, a host, and then in red, a virus, or for example, an archaeal host, and an archaeal cell, and in red, a bacterial cell, what could happen during evolution is that eventually the red Darwinian individual getting tighter and tighter in terms of integration with the black individual will eventually lose its autonomy to replicate independently. It will abdicate, if you wish, some sort of evolutionary autonomy. It will, in that sense, no longer be Darwinian individual. It will de-Darwinized. And now the red component needs the collective to replicate. But that's not very easy to put a virus inside a host. That's not very easy to put a bacteria inside an archaea without policing mechanism. So my very important point here is that it is possible that during evolution, within a given Darwinian individual, that is a collective, enters components that have de-Darwinized, and these components are maintained in the collective by some policing mechanism. The question is, does this hold during aging? This is a very smart quote to say I didn't make up that word. But what is happening during aging in this kind of Darwinian individuals? What has been observed empirically is that endoviruses reactivate with age. Retrotransposon reactivate with age. So if you wish, those components that were initially able to replicate independently, but then were part of this collective, they are now somehow regaining some replicative autonomy. So maybe that's X de-Darwinification or something different. But the only point I wanted to make here is that this is paradoxical because during aging, the individual seems to be divided. And in theory, by etymologically speaking, an individual should not divide, but aging does that. So there is room for philosophical questions here. Then there is a third and still related ontological question that I think is a gray area of the theories of aging, and it is what is happening to Darwinian individuals during what I call hyper-aging, when some organisms live very, very, very long, to the point that it seems unlikely that natural selection is directly responsible for their persistence at very, very old age. If you are 117, it is unlikely that you come from a long lineage of people who experience this specific genetic background that you have. And so it's not because natural selection is keeping you alive that you are alive at this age. So what is keeping you alive when you are that old? And an alternative explanation could be possibly of a systemic nature, because we know that the architecture of a system is critical for the robustness, for the persistence, even for the resilience of a system. So for example, if you have here a system that is in series and a system that is in parallel, you know that if one of the bulb breaks in the system in series at the end of the system. If one of the both breaks in the system in parallel, that's not quite the end of the system. So the architecture of a system may explain how some systems last longer than others, even if this is not under the direct supervision of natural selection. And so to explain the very, very long life, I think we could have, in ontological terms, two main hypotheses. First, that the hyper-aged organisms are still Darwinian individuals, that they haven't lost any essential properties that were making them Darwinian individuals, and so there is nothing to worry about. Or that maybe this hyper-aged individual could have their persistence explained by some systemic feature, and in that sense, that they have become somehow topological individuals. So if we take this hypothesis seriously, that would imply that the ontology of aging organisms could be more complicated than assumed. Because in the standard theory, by default, also this is not discussed, it is considered that basically you are born a Darwinian individual and you will die a Darwinian individual. But I think if you live a very long life and you are no longer able to reproduce, you have lost one of the essential features of what usually characterizes a Darwinian individual in our population. And so maybe that's just a matter of degree, and as time goes by, the Darwinian individual losing functions are maybe a bit less of Darwinian individuals, or maybe they are Darwinian individuals reduced to survival, or maybe they have now reorganized, in a sense, restructured their biology. They're experiencing a new biology, a different biology that, for some topological reason, is sufficiently stable for them not to die. And if you believe in that, then this topological individual could have two different kinds of impacts, at least, on their environment. There could be topological individuals that basically do less of the things that the youngest individual were doing, and so they would have a lesser ecological impact, but they would not be dramatically different. I call them the ghost. Or they could be some topological individual that make a very different impact on the ecology of the planet, and because they change, if you wish, of alimentary regime, so I call them the zombie. And then in the late future, there could be also individuals that would persist as part of collectives that would be supporting them, for example, biotechnological collective, and they would be augmented individuals. Or if they don't direct this collective through their brain and will, they may be more seen as organs of machines that are keeping them alive. So just to say that there could be a complicated ontology of aging organisms to be developed. And now I move on to the final part of the talk, and I focus more on the systemic areas of the current theories of aging. These theories, I said, are focused on organisms, and so many of the metaphors are really not very systemic in nature. But if we adopt more systemic models of aging, then we can maybe uncover additional sources of aging and additional explanation of aging. The first case I will tell you about is about maladaptive aging, and the second case I will be telling you about is how there could be a diffusion of aging across interconnected systems. So let's start with maladaptive aging and what I call phylogenetic antagonistic pleiotropy. I said before that present-day organisms have some networks in them that contribute to their aging, and I think that's acceptable, but that these networks evolved through a long history, the natural history I was mentioning before. But this history is not without systemic constraint, without architectural constraint, because the networks in this series come from former networks, and they were probably not born from scratch. For example, you could have at T0 a network all in black, and at T1 you can have an evolved version of the network, possibly with new interaction between old nodes. Those are the red edges between the black nodes. Or maybe you can have an expanded network with new gene families, new proteins being evolved at time T1 and now connected by the blue edges to the former network. What I'm saying here is simply that we can have what I call an evosystemic vision of the evolution of this network. We can look how the structure changed, and we can also look how the component, the nodes, and the edges change in these networks. And interestingly, if you have an old cell protein that have been for a long time in an evolving lineage, maybe you have a time to make many, many connections with many, many neighbors. And at some point, if you have made many, many connections, you have become very central in the biology of your lineage. But if you have made many, many, many connections, your evolution possibly is quite constrained because it's difficult maybe for you, old node with very many connections involved in many processes to evolve very freely. And it may be possible, maybe difficult for you, old node very central, to welcome new connections with new gene families or new proteins without breaking down very essential part of the biology of the organism. If you look at the protein mTOR, that is very central precisely in many phenomena associated with aging, and that is the big node in green here, we investigated, and I think that is in humans, but I may be wrong, when the proteins that are interacting with mTORs appeared evolved, presuming that maybe this was a proxy of the edge of the interaction between mTOR and these proteins. And you should read this plot as a clock from the red node that is early cellular life, and then you go down to the clock, and then you move to the very recently evolved gene families. So if you do that, you see the green proteins are neighbor of mTOR that evolved at the time of eukaryotes, so 1.5 billion years ago. And basically, if you read this clock, what you notice is that there are very few, in proportion, connections with recently evolved proteins and more connection with anciently evolved proteins. And that precisely is a model of a reduced evolvability of some ancient nodes in networks that, like I was saying, admitted many, many, many connections to the point of saturation where they become part of rigid, evolutionary rigid subnetworks. And that I call phylogenetic antagonistic pleiotropy because those nodes that were very good for members at the beginning of a lineage early on may become detrimental for the descendant of the lineage later on if the environment changes, for example, and you are in a rigid subnetwork. It's very difficult to adjust a rigid subnetwork to a changing environment. So there may be this kind of problem in evolution. And if that is the case, then the genes that are now part of a rigidified subnetwork may become what we could say aging genes because they are no longer able to keep up by evolving with the selective pressure on them. They are outdated in a way and contribute to the demise of the organism in new environmental conditions. And the joke here is that, for example, the opposite sum, a very good invention early on for our lineage, but if it kills the planet by making all terrible ecological things, then maybe that's good at first for the lineage, but later that's phylogenetic antagonistic pleiotropy. But why is it cool here? It's because in classic theories of the evolution of aging, aging is non-adaptive. But for people who believe in program aging, aging is adaptive. But there is a third way, and the third way is that aging could be maladaptive, the result of systems that are unable to keep up with changes. And that was, by the way, an intuition of Stephen Jay Gould a long time ago when he mentioned the fact that perhaps genetic systems age in the sense of becoming less forgiving of major restructuring. So why would it matter to have this kind of evo-systemic explanation of aging in our theories? It would matter because this explanation relies on different principles than the classic evolutionary explanation. They are not grounded in population genetics here. They are also grounded in topological constraint. And so they tell us about something very fundamental, but also very concrete regarding the natural history of the mechanism processes associated with aging. And they support unusual questions. For example, where do aging genes come from? Well, it may be that aging genes, some of them come from architectural constraint that solidified, rigidified the network. Finally, the pioneering work of Annette Bowdish, a biodemographer that I mentioned earlier, is also asking us about aging in interconnected systems across scales and asking them to introduce this kind of modeling in our models of aging. So what Annette does is that she is trying to evaluate the risk of dying or the risk of disappearing with time of structure that are higher than the organismal level. Couples, families, households, societies, do they see the risk of dying increasing with time? But she also asks a question below the organismal level. Cells, organelles, genes, molecules, do they have the risk of dying also increasing with time? And if we frame this in terms of network, is the case that the molecular networks from which the organism emerge have a risk of dying that increases with time. You know, the interactions that are making the network, they could be born in young organism, or they could be born in old organism. Is it the case that interactions that are born in young organism last longer, have a lesser risk of dying than the interactions that are invented late in our life? If that were the case, it would be very difficult for us to live forever because we could not create stable, robust networks. By contrast, if our inner networks were not showing any variation in the risk of dying with time, maybe we could live endlessly because we are not going to be let down by our molecular systems. This kind of question we can investigate using network analysis and precisely computing the risk of dying of interaction within networks. So we go to molecular networks during the lifespan of organism and we say what interaction, what cohort of interaction have a risk of disappearing with time. But we could do the same within organism, within ecosystems. We could wonder whether the ecological ecosystem to which we belong have a risk of disappearing with time that increases, is stable, or decreases. And it's not the same for us because if we are aging in an aging community, then our fate is pretty compromised. But Ulanovich has proposed, for example, is an ecologist that indeed ecosystems are likely to age by rigidification. So it's not a totally foreign thought. But what I wanted to say is that if this is the case, that our inner system are affecting how the organism age, and that when organism age, it affects the stability of the ecosystem to which they belong. And if when the ecosystem age, it affects the aging of the organism, which affect, or this diachronically, the aging and stability of its molecular network, then we see that we could use models that are much more complicated with eventually diffusion of aging through interconnected systems with feedback components and relationship. So the bad joke would be that we are aging in a terrible way in an aging environment, but maybe as age distorters do, we contribute to age our own ecosystems. So that's a curious question, or maybe not a curious question. And there are, of course, many other ethical issues that we could think about when discussing aging and gray areas. Should we fight aging in poor health? And if so, what approaches are acceptable? What are the issues of transhumanism and aging? What are the issues with being racist against aged people? It's not the topic of my talk today. I just want to say that I believe, after having read quite a few things, that if we align human interest, social interest, ecological interest, we might be aging in a different way than we do age now, and that there may be good things to do to our environment and social inequalities to age in a better way. So to conclude my talk today, I wanted to say that it's timely to work on an expanded view of the causes of aging. And I proposed an epistemic strategy to do this. I said, maybe it's time to move from these models that were considering aging centered on organisms from population of a single species to dive into these developmental, phylogenetic, eco-evolutionary, systemic, ontological gray areas, and to move towards models that are more systemic in nature, multi-scale, with ecological interaction and eventually propagation of aging across systems. What I think is very important to point out is that tools and data are available today to do this. We can actually investigate age distortion. We can investigate co-evolution of aging, we can investigate post-selection shadow, we can investigate the evo-systemic of aging, and also the demographic aging of system. I think, of course, this will ask plenty exciting philosophical question, because if we move from the model to the left to the model to the right, then there is a lot of work for philosophers. I also believe that when I look at this picture, that aging occurs at crossroads of many kind of interactions and has many, many causes, which in a way is interesting because it suggests that we can pull on many different strings if we wanted to change the course of our aging. But also it says that because there are so many open issues about aging and so many gray areas that we should be careful about our preconceived notion regarding how, when, and why organisms age, because I believe we don't understand that very well. With that, I will soon conclude. And first, I need to try to make money here, so I will do this. Please buy my book. It's a very good book. Also, it has a terrible rating by a hater, so buy it and put it good grade, please. Second, I'm also self-editing because I'm really striving for money. Collection of books on evolutionary biology, microbiology, and biology of aging for kids that you can find on the internet. And of course, I want to thank very much all my wonderful colleagues who helped me daily in the lab to do all the work. Phil Lopez, Eduardo Corel, Philippe Hunman, with whom I collaborate, Yuping Dai, Annette Baudisch, for Doolittle, Jonas Martins, Frédéric Bouchard, who inspired some of my work, Beshish Batachea, Jérôme Tellière, François-Joseph Lapointe, Raphaël Ponce Toledo, Juliette Jasinski, who started in the lab. And I'm very open to collaboration with whomever is nice and want to collaborate with me, especially if you're rich. Thank you very much for your attention. So I can take questions, but I don't know to use on the screen. Oh, thank you. Thank you, sir.
And we don't have much time. So short questions, short answers, please. And speak loud, please.
Oh, yeah.
Because I think the microphone has to, or maybe come closer.
Oh.
Is it okay? Thank you.
Am I audible?
Mm-hmm.
This is a very okay. I was just wondering if, like when you were talking about age distorters, I was wondering if it's like evolutionary advantages for parasites and symbionts. I was wondering why they haven't evolved more robust systemic mechanisms then to kind of detect and neutralize these manipulations. And if in a way it kind of suggests that age distortion is evolutionarily stable, I was wondering if it's because aligns with, you know, like the host's life history trade-off, or if it's more of like an evolutionary arms race. Yeah,
thank you very much for your question. A short answer, I don't know. If you want a slightly more developed answers, I think there could be different situations. But first, we have to remember that age distorters are a hypothesis, and that and there are different types of age distorters. I didn't describe that here. A type that is directly interfering with these host pathways that are involved in host aging, and a type that is basically interfering with the amount of damages in the host body. And in papers, we distinguish these two types. But also there are distorters who could slow down the aging of the host. And we would be happy to actually have a healthier host who would live longer. So there are, if you wish, good and bad age distorters from our perspective. But it could be also observed in ant colonies where some ants are infected, and I don't remember by what nematode exactly, but then they seem to be living longer, and the workers and infected ants are treated like queens and everything. So it seems their life is perfect. They don't have to go outside as the other workers. They risk their life less, and they stay closer to the center of the colony, of the nest. But then when birds attack, these infected ants that seems to be living a very good life by our standard, and they are the first targets of the birds. So it's complicated, but I think what is very important in the story is that we are not aging alone, and most likely aging organisms are not aging alone. So this kind of inter-species interaction interferences should be modeled explicitly.
Yeah. Just talk about that. Si oui, on peut better.
Ah, non, ce sera le max.
So thank you for your talk. I wanted to know, there are like now some concepts like extended immunology and extended defense, and is your concept of extended aging similar to them, using the same mechanisms, or is it just different?
I must say I don't know the concept of extended immunology, or you have to teach me. That's my answer.
Oh.
Thank you very much for the presentation. On this like eco-evolutionary aspects, I was wondering if it would also impact the way we actually measure aging, like with the epigenetic clocks and so on, because so far I've seen that showed that, for instance, the HIV virus can actually like modify these markers, but should we also like think of refining them in some ways with this new perspective?
Yeah, it's a very difficult question you're asking, because measuring aging, we don't know how to define aging, and we're not sure there would be a unique measure and what measure to take. But I think, bearing this huge limit in mind, it is very true that it could be useful to quantify other aspects of aging than the one we're currently quantifying, to take into account this socio-environmental impacts on aging. And a simple model would be a gauge, if it's proper English. How often did you meet age distorters, for example? And if you are under a certain threshold, that's fine. But if you have met them too often, then you're maybe going to age faster. So what could be quantified here is how high on the gauge you are of terrible encounters, for example. And that would be a form of a measure of social aging, if you wish, but that would be different from biological aging. So yes, there is room for new measure and measure of different processes that could eventually potentiate biological aging. And so my short answer is yes, but it's difficult.
Very general question. Can I ask it? So, thank you very much. Fascinating talk. I thought aging was a mess, and now I think aging is a mess. And then you showed that it's even more messy than we think. There are more interesting questions, and you showed that very well. So the questions are fascinating. My practical question is, what makes you think that by adding those fascinating questions, you will really in the end clarify the issues that you presented in the first place? And I was thinking about historical precedents. Do we have examples where by adding more legitimate complexity, because this is what you're doing, we have clarified an issue? I mean, is that something that you've investigated? And do you think that it's promising to do this, and why? So it's a very general question about your strategy. I think it's a reasonable question given that now I'm even more confused than I was in the first place.
Yeah, it's a very good question. I think it's probably an act of faith here to decide that embracing complexity is valuable. So that's maybe my preconception. Maybe I was raised with thinking in this way. And not being an historian of philosophy, I'm probably not able to tell that, yes, it made a big difference to be able to welcome complexity to better explain some phenomena. But out of the blue, I would say, try to explain diseases without discovering microbes. You're probably not going to do a good job, you know. Try to explain antibiotic drug resistance without lateral gene transfer. It's probably not going to work very well either. So in general, I think as a realist here, that it's, even though all this is models, it's probably not the worst road to go to try to embrace complexity. And then, if it's too complicated, then simplify again. But I think if we start with overly simple models in the hope of understanding complexity, we may not be using the best strategy.
You should come here. You cannot do an exception. Sorry, my bad. Even though you're old.
Is there any strong reason why you would call an age distorter and not just an age modifier or something like that? Because distorter does seem to suggest that there is something like non-distorted aging, and what would that be then in that case?
It's a very good question. The first reason why I called age distorters was because of the sex distorters. And that's a notion that evolutionary biology can be comfortable with. So there is a bit of a rhetoric trick, if you wish, in that. Say, oh, you used to think about sex distortion. What about age distortion? And so that's how I came up with that wording. But indeed, I believe that no one ever aged alone. And for that reason, it seems very difficult to consider that there is a normal aging. Also, we can enter into a statistical discourse, and we could say, that in statistical term, that is average way of aging or the most consensual way of aging, and then we can quantify departure from this consensual mode of aging. So it's still something that could be empirically discussed to what extent age distorters distort consensual aging.
And also, I mean, just a follow-up question maybe on that. There's also a question of, there's also a question of, I would say, how would you measure that? What would you measure the progression of in that case? Like, for instance, if you have a distorter that is changing the way your, let's say, biology was working in one tissue, in one organism, would you measure something in that tissue, or would you measure, I don't know, risk of death at the level of the individual organism or something else?
Everything. And that's not a joke as an answer, because what you want to know is how the distorter reorganizes the progression of the life of the organism. So if you can use transcriptomes, for example, you can test in case you meet or didn't meet an age distorter, whether your biology has been reoriented toward different kind of processes, and you can do that with a rather holistic view on the processes that are being affected. You don't need to have a priori one clock or one measure. So you could do this in this way, and that would be the way I would prefer to use, because then I could learn broadly about the many ways to progress toward death in aging organism as they meet age distorters. But you could also very simply quantify it using a phylogenetic clock if your organism is amenable to this kind of study, and say, oh, it seems that your biological age by this clock is now 5 years younger or 10 years older. Or you could, like you said, quantify how exceedingly fast you end up dying, for example.
So you could also have conflicting effects of age distorters.
Yes, you could. Yeah. And I'm wondering whether in our microbiomes, for example, we host some age distorters with conflicting effects on our survival, with some bacteria doing us good in our old days and others not.
Okay. Thank you.
Thank you.
I think it's probably time now. Yes. Yes, of course you can.
So it's very plausible to say that these standard theories of aging cannot be applied to unicellular organisms. But I was wondering if one can still try to push a bit further to apply them. Like, it seems to me something like a generalized version of the disposable soma theory might be applied to unicellular organisms, by saying there is a division of labor between growth and reproduction, for example, or maintenance and reproduction. So it doesn't require that to be in different cells. And I was even wondering if maybe, when you showed the example of the yeast cells that accumulate scars on its membranes, if maybe you might even have something like antagonistic biotropy in unicellular organisms.
So those are very good questions and certainly difficult to address. You will remind me the one I would forget. I will start, I think, backward. If you look in the literature whether people have been willing and spontaneously expanding, for example, disposable soma to non-animals? The answer is no. So only one instance, it was the case of the yeast, the budding yeast, because it has a big growing mother cell and very obvious asymmetric cell division with a small daughter cell. It has been suggested that maybe that could be a case of soma versus German kind of division, and maybe with a sacrifice of one cell to the benefit of the other. But in general, people haven't done this kind of exercise of expanding the scope of one theory and suggesting that it would apply at a much broader phylogenetic scale. It is difficult to do, and that would answer for antagonistic pleiotropic theories when the growth and the reproduction cannot be distinguished, whether an organism is growing or whether it is reproducing. Whether you can't decide between the two, it's very difficult to imagine a trade-off then, and what trade-off would be occurring. And for that reason, it's difficult to imagine to apply these theories to microorganisms in a straightforward way. Plus, when would selection weakens during the cell cycle? It's not very obvious,
on the level
but
of cell
you,
cycle, but
yeah.
no longer like considering.
Oh.
Maybe we can discuss about this.
Sure. But anyway, so the short answer is very difficult. And so there are two roads. Either we expand the existing theories and we explain why they could work in a broader framework, or we create new theories. Yeah, more encompassing ones. Thank you.
Thank you very much, everyone. Thank you again, Eric, for the fascinating talk. And Thomas, maybe you want to announce the next.
No, because I don't remember.
Ah.
Rick Michaud in January, but I don't know the exact date.
How could you forget?
I mean, I know the name. I don't remember the date.
Okay.
But it's mid-January.
Okay. Thank you again.